the 16th edition of our INFOGEST Viva webinar. My name is Farhad Garavand, and I would like to introduce Daniela Freitas, who will conduct the webinar with me today. The event is organized by a team of postdocs from Teagasc Moorepark Food Research Center here in Ireland. These webinars aim to share the latest findings in digestion with presentations from the most recent graduated PhDs, postdocs, and young researchers. This webinar is broadcast live on YouTube and will be accessible on the INFOGEST YouTube channel. The presentation in a pre-recorded video will be followed by a Q&A session with the speaker. So I actively invite you to use the Q&A button at the bottom of your screen to ask your questions. Our speaker today is Charlotte Oliver with in vitro assessment of milk extracellular vesicles, digestion and bioactivity. Charlotte is now working in H&H Group as a researcher here in Ireland. She recently obtained her PhD in food science at Trinity College Dublin. There, she performed a comprehensive characterization of in vitro assessment of milk extracellular vesicles, their resistance to digestion, and their bioactivity. Her project is looking at bovine milk extracellular vesicles to see their applications in infant milk formula production. So Charlotte, the floor is yours.
Hi everyone, I'm Charlotte Oliver, and today I'll be presenting the work done during my PhD, which looked at in vitro assessment of milk extracellular vesicles with a focus on their digestion and bioactivity. I was supervised by Linda Giblin and Andre Brodkorb in Teagasc, as well as Lorraine O'Driscoll in Trinity College Dublin. So overall, the aims of my project was to see the effect of bile and enzyme digestion on EV's recovery, as well as their effect of EVs on healthy adults and an infant gut barrier. And finally, see the effect of EVs on downstream target cells. So look at EVs and bioavailable EVs, so the basolateral compartment of CACO2 cells. See their effect on muscle and fat cells. So we wanted to understand the health effect of EVs to see if there is a nutritional interest of supplementing infant formula with EVs. First of all, so what is milk? Very general slide. So milk is the primary source of nutrition for the newborn. So it's a complex matrix with water, lactose, fat, protein, but it also contains immunoglobulin, enzymes, growth factor, antibacterial agents, and as well it contains extracellular vesicles. And interestingly, milk and also extracellular vesicles composition change based on the animal species, the stage of lactation, or the health status. So extracellulars or EVs, if you're not familiar with them, there's small particles, basically a little sac with a lipid bilayer on their surface. They also contain protein that are on their surface and as well contain inside the EVs. They're basically also carrying nucleic acid, and their function is to bring message to target cells. So they're secreted by all cell type, and that's a way for the organism to transmit message. So to study them, there is an organization called International Society of Extracellular Vesicles, and they released some guidelines to help researchers work with EVs. And in the latest one, in 2023, they actually provided some guidelines for the first time on how to work with milk extracellular vesicles, and provide information on actually how to obtain EVs. So first of all, I wanted to know when I started my project, what is the concentration of EVs in different milk? So the concentration varies greatly depending on the method used to measure the concentration, but it's around 10 to the power of 9 to 10 to the power of 12 in human milk, while it seems to be higher in bovine milk with concentration going as high as 10 to the power of 13 particles per milliliters. And interestingly, in infant milk formula, they were detected. However, they appeared damaged, and their concentration was also significantly reduced compared to skim milk. And the interest of extracellular vesicles is that they could promote gut barrier health. So a lot of studies have come over the past 2 or 3 years on the role of EVs on inflammatory bowel disease, on how in vivo they help actually reduce the symptom intensity. In addition, they could actually promote the immune system maturation and also help muscle protein synthesis. But since their concentration is reduced in infant milk formula, our question was, should we actually add them to infant milk formula? Would that provide a health benefits to the formula-fed infants? So during my PhD, the gaps of knowledge I tried to answer were, first of all, in the field of EVs and digestion. So we know that EVs, or it seems that EVs survive digestion, but we don't know the concentration of EVs after gastrointestinal digestion of milk. Additionally, we don't know the effect of bile treatment and enzyme digestion of milk on milk EV concentration. And finally, there was little focus on, so bovine milk, the primary source of milk in infant milk formula, and infant digestion and EV recovery. So when it came to the gut, so the effect on EVs on paracellular permeability and gut health on a healthy gut is not really characterized. And also we wonder, so for the infant, could they help promote gut maturity? And finally, on downstream target cells, so what are the effects of bioavailable EVs? So what happened after they're actually absorbed by intestinal cells? And what's their effect on target cells, so such as myotube or adipocytes? So I chose those cells because in infants actually go through a high muscle protein synthesis and also important adipogenesis. So in my first research chapter, I track the fate of EVs during infant digestion. So to do so, as all of you may be aware, it's not possible to study it in vivo and particularly not in humans. So hence the INFOGEST network, and in 2018, the paper published by Menard et al. Proposing static infant digestion. So we use that protocol and we get our work published this year in Molecular Nutrition and Food Research. So to see the effect of enzyme digestion and bile treatment of milk on EV recoveries, we performed different digestions. So first, we tested the effect of no digestion as a control. We also perform a gastric digestion to see the effect of the gastric phase on EV recovery, a whole gastrointestinal digestion with gastric and intestinal enzymes. We also wanted to see the effect of just enzymes, the effect of bile. And since bile is a surfactant, we hypothesized that it could actually disrupt the EVs that have lipids on their surface. So we tried a lower concentration of bile. So here is just to give a bit of context on how the work was done. Is unfortunately, when you work with EVs, there is no proper consensus method to this day to actually collect good EVs. So you always have to choose if you want to get a high yield or a high purity. So you choose your methods based on that. So we used two methods. The first one was ultracentrifugation, so where we basically remove caseins that are a major contaminant when you actually work with EVs because they have the same size and the same density. And we perform a second protocol, which is density gradient. So we know the density of EVs, so basically we separate it based on their density. And we developed several techniques to actually characterize EVs, so such as nanoparticle tracking analysis, so to get an information on their size and concentration, atomic force microscopy to get information on their surface topography, and SDS page and immunoblotting to see specific EV markers for proteins. So first we wanted to know if we could detect some EV protein markers after digestions. And so what we observed is, interestingly, so after gastrointestinal digestion, so there was a significant reduce in the expression of the 4 EV markers. So, and when we did just enzyme digestion, 3 out of the 4 marker expression were reduced, while when we did bile digestion, only 1 marker was reduced. And when we increased the bile concentration, the concentration was reestablished. So, based on this observation that enzyme reduced the expression of more markers than bile, we suspected that enzyme had a greater effect on EVs, and also that a portion of EVs are actually damaged during digestion. So at that stage, we were wondering if they were even surviving digestion. So next, we looked at actually their particle size and concentration to see if we could detect particles of the size of EVs. And so what we observed is that so we could detect in all of our samples particles of the size of EVs. And we could also measure some particle concentration. And interestingly, after bile and enzyme digestion, our EV preparation had a lower particle concentration. So this suggests that EVs are still present, but at a reduced concentration after enzyme and enzyme digestion. So, but to confirm their presence, we finally looked at EVs under atomic force microscope, and we could observe them. So before digestion and at the end of digestion, however, their surface appear damaged. So that led us to the conclusion that EV yield is decreased after intestinal digestion, and that enzymes appear to have a greater effect compared to bile. However, some EVs were still present at the end of digestion. So since there were still some intact EVs, we wanted to know then, we were sure that some EVs were actually reaching the gut. We wanted to see what were their effect on a gut barrier model. So we used CACO2 monolayers where we actually applied EVs and we monitored barrier integrity by measuring paracellular transport, permeability, inflammation, and we looked also at tight junction proteins, which allow cells to actually attach to each other, preventing also some molecule to translocate and reach the bloodstream. So I am not showing some results here for because I don't have the time, but basically we didn't see an effect of EVs on inflammation or on transepithelial electrical resistance or paracellular permeability, but interestingly, we saw that EVs could modulate 2 of the 3 markers we tested for tight junction. So they were able to modulate claudin and auclidin, which are so involved in charge selectivity of tight junction, so controlling the charge of the molecule going between the cells. So that was their main effect on a healthy gut. So then we actually tested them on an infant model. So this infant model was actually developed in our lab, and this infant gut is a sodium butyrate treated gut, so that this treatment actually increased permeability while not inducing inflammation. And so here, this is a nice graph just explaining that after sodium butyrate treatment, basically the tear was decreased even after 24 hours, although there was a slight increase, showing that the cell could recover. So then, once we saw that the cells could recover, what we did is we actually let the cells recover after basically the treatment that made them leaky. We wanted to see if they could recover with EVs. And as a positive control, we use L-glutamine, which is known to strengthen tight junction and promote strengthen the gut barrier. And so we saw that our model recover naturally after 72 hours of treatment, and after 24 hours of treatment, the monolayer actually, so EVs and L-glutamine help the monolayer recover. So those data, those two results, so the modulation of tight junction and the increase of tear after sodium butyrate treatment shows that EVs can modulate actually so healthy gut monolayer in vitro, so that they don't have just effect on inflamed gut as it was previously demonstrated in the literature. And so finally, the last chapter we wanted to see, so we work with our CACO2s, so they're on a transwell, and basically you have an apical compartment with the cells, and they're on some sort of, I'd say, basket, and basically what goes in the basket is cell secretion. And so the basolateral are the bioavailable, so what is going to reach the bloodstream. So we tested our intact EVs and our bioavailable or basolateral EVs on target cells, so where we tested myotubes, where we looked at muscle protein synthesis and muscle cell differentiation. And for adipocytes, we looked at adipogenesis and adipolysis. So the first experiment I did was I looked at a marker involved in muscle cell differentiation, which is myogenin. And interestingly, so EVs after 24 hours of treatment significantly increase myogenin concentration in cells, so showing that it helps promote muscle cell differentiation. However, basolateral EV had no effect. So we went and checked that there was actually EVs in the basolateral, in the bioavailable samples, and we observed a significant increase in particle concentration, so suggesting that actually EVs are present in the basolateral compartment. So then we looked at markers of muscle protein synthesis in muscle cells, so which are one pathway, which is the mTOR pathway, which when it's phosphorylated, lead to the phosphorylation of other target protein, leading to muscle protein synthesis in the nucleus of muscle cells. So we looked at mTOR phosphorylation and its target protein P70-SSK. So interestingly, EVs increased mTOR phosphorylation while basolateral EV had no effect. And even more interestingly, we found that for P70-SSK, basolateral EVs could have an effect, but EVs had no effect. So these results suggest that actually basolateral EVs have a concentration, and that even a low concentration of EVs can modulate target cells, at least in vitro. And so then we looked at their effect on adipolysis. So here what I did is I differentiated my immature adipocyte with the samples for 8 days, and I looked at the fat droplet formation. So I stained the fat inside the cells with oil red, and as you can see, EV's fat droplets were significantly lower compared to the control and closer to the level of the negative control, which was resveratrol. However, there was no difference in basolateral EVs. And finally, I looked at glycerol. So release in the model for adipolysis. So here, I differentiated my immature adipocyte into mature adipocyte, and then I treated my samples for 24 hours with EVs or basolateral EVs, and I observed that EVs significantly increased the release of glycerol, so promoted adipolysis, while basolateral EV had no effect. So showing that overall, so EVs were able to modulate myotube and adipocytes. However, the concentration in basolateral might be a bit too low to have an effect. So overall, the take-home messages from my presentation are that EVs are resistant to gastrointestinal digestion, that intact and damaged EVs are reaching enterocytes, that their effect seems to be in the gut. However, the concentration of EVs reaching muscle and fat cell in vitro may not be high enough for them to exert an effect. So based on my results, I would recommend to add EVs into infant milk formula. So that's, let's say, I would not recommend, but that's a very good first preliminary step. However, so now the next question remains is that it's very difficult to produce EVs at a large scale, so there is no proper method. And so should we use, I don't know, more analytical method? And so also when should it be added into infant milk formula, since also EVs are heat sensitive? So should they be added sterile after heat treatment? Should they be encapsulated, but that'd be costly? So that's a talk for another day, I suppose. So thank you all for listening to me, and I'd be happy to answer any question.
Hey, thanks very much, Charlotte, for such a nice presentation. You did a really extensive work during your PhD studying the composition, the EV content of milk, bovine milk. You mentioned at the start of your presentation that one of the differences between bovine and human milk was the concentration of extracellular vesicles. Would there be other differences that you would point out in terms maybe of functionality, bioactivity of the vesicles, extracellular vesicles present in human milk compared to bovine milk?
Thanks, Daniela, for the question. That's a very interesting question. So, as you said, so the main difference we know so far, let's say, is that their concentration is higher, seems to be higher in bovine milk. So when it comes to their composition, if you look into in-depth composition, you might see difference. You know, let's say if you look at the whole proteomic or the whole microRNA composition, and in terms of functionality, so that's let's say that we're discovering every day. So both of them seems to have an effect in the gut, and especially when it comes to colitis and reducing the severity of colitis in animals. And one very interesting study that was published last year mentioned a difference in muscle in vitro and in vivo, that actually human milk EVs could promote more muscle protein synthesis than bovine milk at the same amount, interestingly.
Thank you very much. Let me see, we have one question here. Would bovine and human extracellular vesicles work the same in infant formula? Would it also be advantageous for adults?
That's a good question once again. So it's possible that, you know, the EVs you add in infant milk formula would be heat treated, so potentially damaged. So so they could actually be different. And so the second part of the question, I believe, was so would it be advantageous for the adult? That's possible as well. That's also kind of understudied, but that's what our in vitro gut work and let's say cell muscle and fat cells seems to demonstrate that it would also have a benefit on adults. But let's say for adults, usually if you consume, bovine milk or any milk type, I don't know, goat milk, you're gonna get those vesicles. While infants, like let's say below 6 months old, would only get either human milk, so they would get EVs, but the ones that are formula fed would actually get damaged EVs.
I think there is one more question. Okay, so the question is, what is the best method, or what's the method used to estimate EV concentration in bovine and breast milk?
Sorry, I was muted. So the method we used is called nanoparticle tracking analysis. So basically, it's a technique based on the Brownian motion of particles. So if some of you may be familiar with, techniques like master sizer, zeta sizer. So basically, you check where the light of the particle is scattered, but let's say the nanoparticle tracking analysis has another, so it gives you an information on the size based on this, but it also has a camera. So basically each particle that is scattering light, the machine can actually count and give you a concentration based on that. And based on that, then we know, the amount of EVs, and we actually report it per milliliter of milk, based on dilution or preparation step. Other methods, so you have other methods that I didn't work with during my PhD, but you could use, let's say, flow cytometry could also give you an information with number, but it has a major limitation is that EVs are actually at the threshold sides of the flow cytometer, so they're a bit small. So they have, you have a lot of artifact in your counting. So that's some of the methods that, let's say, I'm familiar with in terms of counting EVs, but there are other methods that I'm a bit less familiar with.
Sorry, I was on mute. We have 3 more questions that just came up, but maybe we won't have time for all of them. Maybe for the first, since we were talking about methods to test or isolate extracellular vesicles, were you able to observe or test EVs isolated by density gradient?
I could actually observe and test EVs isolated by density gradient. So my project was a collaboration between Teagasc and Trinity College Dublin, so in Lorraine O'Driscoll's lab. And basically, so in Teagasc, we have big expertise on digestion and milk, and they have big expertise on EVs, but not milk. So they established different methods, and they observed that actually the density gradient was the one giving the highest purity, because they compared different methods, and they observed, usually the way they also saw it is by observing by transmission electron microscopy. And I saw a question about the caseins. So that's quite interesting. The caseins, basically, so they cause a problem, when you work with EVs, because they're the same size and the same density. But what we ended up doing is removing them using, like hydrochloric acid, basically, to precipitate them. So then you can actually increase your yield and your concentration of EVs, and caseins are basically less of a problem then.
Charlotte, maybe one last question. Can EVs also be formed by milk-producing cell lines? Is it studied? Do you know if this is being studied at the moment?
So that's exactly it. So since all cell types secrete EVs, so we don't know exactly where the EVs are coming from in milk, but it's highly likely that they're secreted by cells in the mammary gland. So then I would not be able to tell you, which cells in the mammary gland account for which EVs, but definitely the cells are the cells from the cow or from the mammary gland are the one producing the EVs.
Okay, thanks. Thanks very much, Charlotte. We do have a couple more questions, but maybe we will type the answer in the chat because we have to end the webinar. It's time to end the webinar now. Thank you very much, Charlotte, for presenting all the work you did during, or not all the work, but for summarizing all the work you did during your PhD. It was a pleasure having you here today. Our next webinar will be on the 6th of November, and we will have a presentation by Maria Santiago-Vivanco from the Institute of Food Science Research in Spain. So stay tuned and see you then. Thank you for attending.
Thanks for having me. It's been a pleasure.
Thank you.
Thank you. Bye.
See you next month.